Now we're in the phylum Chordata, which includes tunicates, lancelets, hags, lampreys, sharks, all the bony fishes, amphibians, reptiles, birds, and mammals. And this is the big and most successful group of deuterostomes. So chordates have some unique features. There are four basic chordate features. There are a few others also, but there are the kind of the four major ones. A notochord, which is a stiffening rod, and you can see it labeled in this little drawing down here. It's not a backbone. It's a kind of a stiffening rod. It basically provides something for muscles to pull against on each side of the body as the animal is kind of has a swimming motion, a side-to-side swimming motion. There's a single dorsal hollow nerve cord, and remember, it's dorsal. That nerve cord is dorsal to the notochord. It's closer to the back than the notochord is. And you only have tiny remnants of the notochord left, but you still have a single dorsal hollow nerve cord. If you look at a lot of the invertebrates, they have ventral nerve cords. Often they're paired, they're not single, and they're usually solid, they're not hollow. So there are some differences there. Pharyngeal slits are really important. The pharynx is inside the mouth, that first length once something has moved inside the mouth, and there are these slits. You will often see them incorrectly called gill slits, labeled gill slits in diagrams. They are not gill slits. In a lot of chordates, they have been modified into gill slits, but they are not gill slits. They did not evolve as gill slits. And then there's a postanal tail. So if you think of an animal like an earthworm, the end of the digestive tract is right at the tip of the animal. There is no postanal tail. But in chordates, at least ancestrally, the digestive tract exited before the back end of the animal. So you had this segment of the animal that had no digestive tract in it. That's the postanal tail. Within the phylum Chordata, the first subphylum we're going to talk about is the subphylum Urochordata. These are tunicates and sea squirts. And these animals generally have a kind of a sponge-like lifestyle. So they have an in-current siphon right there where water enters the animal. And they are marines for the most part, so they're taking in seawater. That water passes through the pharyngeal slits, and you see all these little openings here. This is the pharynx, and those are the pharyngeal slits. And there's a mucus layer there that filters the food particles out of the water. And then cilia make that mucus kind of bring that material down here, because then you're going into the digestive tract right there. And then the clean water exits through the excurrent siphon right here, and that's partly covered up. The label's covered up, but that's the excurrent siphon. So they're just essentially filter feeding, and they are attached to some substrate on the bottom. They're not moving around, at least not as adults. Now, when you look at an animal like this, of those four chordate features, the only one that's really obvious is the pharyngeal slits. They've got lots of pharyngeal slits, but do they have a dorsal hollow nerve cord? Yeah, not really. They have a notochord. Well, not really. Postanal tail. Well, it's hard to say where that would be when you look at an animal like this. So they don't really have those features, but that's only true for the adults. You're looking at an adult here. The juveniles do. Now, I should mention they're called tunicates because they are covered on the outside with a non-living tunic that kind of forms the outer layer of the animal. Here's what tunicates look like in reality. So you're really looking at the tunic here, and you can see the in-current siphon is right there, and there's the excurrent siphon. So water's going in there through those pharyngeal slits and then out through that opening. So this shows the stages of development for a tunicate. So what I showed you, the diagram I just showed you, is basically a version of this. There's the adult form, and again, there's the in-current siphon, those pharyngeal slits, and the excurrent siphon. And of course, there are some other organs in there, but that's the major way that water flows through the animal. And the adults are attached to a substrate. But if we back up to what they look like when they're larvae, here's the larval form, and they actually will call it a tadpole larva because it does look like a tiny, tiny little tadpole. If we look at that animal, well, we've got, here's the incurrent siphon and pharyngeal slits, so that's one of the chordate characteristics. Here is a notochord right there. Here is a single dorsal hollow nerve cord right there, and then there's the postanal tail. So it's the larval form has all of the chordate characteristics. The adult form does not. Now, the larval form actually can swim a little bit. Like I say, they're tiny, so it's not like they're really fighting the currents very much, but they can swim. When they reach a certain stage, they attach to a substrate head down, and actually, you know, there's where they attach, and they start to metamorphose into the adult form, which you see there, and then there's the adult form. So they start to metamorphose, and of course, once they're attached to a substrate, they're not going to move anymore for the rest of their lives. That's it. All right, another subphylum is the Cephalochordata. These are the lanceolets. The old genus name was Amphioxus. It's Brachiostoma now, but Amphioxus has kind of become another common name for these animals. Cephalocordate literally means head chordate. Now, if you look at the animal, look at the picture on the bottom part of this slide, you can see that it looks a lot like a very slim little fish, and they do look a lot like fish. I should point out that you are more closely related to actual fish than this animal is, but it does have fish-like characteristics. Now, um, this this animal is probably pretty similar to what the ancestor for all the chordates was like, you know, in terms of its body plan and how it lived. So you see the diagram up here, it's labeled. Even the adults have all of the chordate characteristics. Now I want you to notice that they're calling these pharyngeal slits gill slits here. That is totally incorrect, because if an animal has gills, it's breathing with those gills, right? That's what it means if you're going to call something a gill. It means it's harvesting oxygen from the water using that structure. These are not gills. This animal does not breathe using those slits. Those are pharyngeal slits. What the animal uses them for is feeding. It's not breathing through them. It's actually breathing through its skin, through its whole body surface, because they're very small and they have a low metabolic rate. And so it's just diffusion of oxygen from the water through the skin. That's how they're breathing. So, like I say, you will see these pharyngeal slits incorrectly called gill slits lots of times. That's not what they are. Like I say, in a lot of animals and a lot of fish, they're modified into gills, but they are not gills. So the way this works is that water enters right here, and it's in the pharynx. The water then passes out of the pharynx through these pharyngeal slits, but it's still inside the body of the animal. You notice that those pharyngeal slits don't come to the outside. The repeating segments that you see in the actual picture down at the bottom are just muscle segments. So the water is still inside the animal's body, but it's now the food particles have been trapped in the mucus that's attached, that lines the pharyngeal slits, and then the food particles come down here, and now they're in the digestive tract. The water comes outside, the clean water, and it leaves the animal right here through that atriopore. That's where the water comes out. Now, that's not the anus. If you follow the food through the intestine, there's the anus, and it has a small postanal tail right there. And of course, it's got the notochord, the dorsal hollow nerve cord, and all of that. So these animals have all the chordate characteristics. So they're at least formally pretty common in shallow seas around the world. They don't have a brain. They have a nerve cord, but there's no brain at the front end of it. They don't have a heart. They don't really need it. You only need a heart and to circulate oxygen and nutrients when you start to get relatively large. They don't have much in the way of sense organs, and they're just filter feeders. So if you look at the picture of the lancelet at the bottom, you notice it's kind of mostly buried in the sand with just the head end poking out. Well, that's how they live. They live in sandy oceans. They bring water in through their mouth, and in the water, the current that runs through them is just being driven by cilia that are beating. So it's a pretty subtle current, but water comes in and they filter food out of it. They're not swimming around like fish. All right, now the last subphylum is Craniata. The old name for Craniata was vertebrata, and you'll still see that used. These are the vertebrates. So the chordates that we've talked about so far are invertebrate chordates. They do not have a backbone. The craniates do now, or at least mostly do. Um, the thing is that not all craniates have a vertebral column. A lot of them do, most of them do, but not all of them. And so calling them vertebrates is a little bit misleading. So what the feature that all the craniates have in common is a cranium. They have a skeletal structure that surrounds the brain and protects the brain. So this is the most diverse group of chordates out there. They have a well-developed brain, at least relatively well-developed, more developed in some than in others. They have all the four chordate features that we talked about, although they get pretty modified so that they're hard to see. And in addition to those chordate features, they have a cranium. But remember that not all have a vertebral column. That's why they're really called craniates. All right, so important vertebrate features. A living endoskeleton. Now, echinoderms also have an endoskeleton. A lot of successful land animals, in fact, most species of successful land animals, the most successful are the arthropods. They don't have a living endoskeleton. They have an exoskeleton. The exoskeleton is a really good body plan if you're not going to be very big, and you have the opportunity to molt it off, because you can't grow if you have an exoskeleton. The only way you can grow is to molt off your old exoskeleton, and while the new one is still soft, you kind of pump yourself up with either air or water, depending on where the animal's living. And now once the new exoskeleton hardens, there's some room in there to grow into. The advantage of an endoskeleton is that you can grow without shedding the skeleton. You can add onto it and grow because it's internal. The nervous system is, again, at least relatively advanced, and vertebrates in general have paired jointed appendages. So those paired jointed appendages are often fins, but they get modified into lots of other things, front legs and hind legs. They're lost in some groups, but the ancestors of those groups had them. Okay, now, one of the questions that's been around for a fairly long time is, what group gave rise to the vertebrates in an evolutionary sense? And what you're looking at in this diagram is Garstang's hypothesis, which has been around for most of the 20th century. And Garstang thought that if you look at the tunicates, you've got this adult form, which is not very vertebrate-like or chordate-like, but the larva really is pretty chordate-like. So what if you had something like a tunicate larva that essentially was able to reproduce in that form? Now, when we call something a larva, you're actually saying that it can't reproduce. The adult form is the reproductive form of whatever animal you're talking about. It's not the larval form. So if you had something that was a tunicate that was able to reproduce while it was still in that tadpole larva form, you're pretty close to a craniate or a vertebrate. And that's called neoteny, retaining juvenile characteristics into reproductive adulthood. So you retain that juvenile body form even though you can reproduce. And so this, what you see down here, this is hypothetical, but this would be a something that's still pretty closely related to tunicates, but is in that larval form but can reproduce. And from there to the earliest craniates that we had in the fossil record, the Ostracoderms, it's really a very short stretch. It doesn't require a lot of imagination to get from one to the other. Now, Garstang was working back in the early part of the 20th century. He didn't have access to, well, some of the fossils that we have access to now, and he also didn't have any access to information from DNA studies. We now know that this hypothesis is wrong. Um, the first the earliest chordates, well, I should say the earliest craniates, were basically already in a form similar to this, and we have some of them in the Burgess Shale fossils, if you've ever heard of that. Also, DNA studies have shown that the tunicates are not the ancestral group to the craniates. So Garstang's hypothesis was around for, like I say, for most of the 20th century, and you will see it come up, but it's almost certainly wrong. The newest evidence doesn't support it. Okay, so let's look at some of these really early craniates. Um, we didn't have much in the way of fossils when the earliest ancestors of the craniates were around, because they had very soft bodies, and soft bodies generally don't fossilize well. Like I say, there are a few places where we do have good fossil deposits from the right time period. So the Burgess Shale is the famous one, but there are a few others. And in those deposits, some of these really soft tissues were preserved pretty well. But for the most part, you need something hard for it to be preserved over, to be fossilized and preserved over long periods of time. So the first vertebrates that we have in the fossil record that are really abundant are the Ostracoderms. Now, Ostracoderms were certainly fish-like. Um, they appeared in the Cambrian. They had the front half of the body was really armored with bones. So you see that in these diagrams. This front half is really seriously bony. These animals had no jaws, though. They had basically a fixed mouth opening, but they didn't have any movable jaws. Now, they could swim. They could certainly swim, but they couldn't really swim very well. They probably were not spending a lot of their time swimming like fish today do. They were probably spending most of their time on the bottom, but they could swim. Um, they there were a few that might have been doing some predation, chasing some little invertebrates down, but they don't have jaws. So for the most part, they were almost certainly living more or less like lancelets. They're filter feeding. They may be sucking muck up from the bottom and getting the food out of it that way. Um, they they were quite a bit bigger than lancelets. These are generally not big animals, but they were quite a bit bigger than lancelets. So they were certainly too big to just use cilia to make water move through that pharynx so that they could filter food out of it. They were, certainly must have been using muscle contractions to make that water move. After the ostracoderms, the first jawed vertebrates appeared, the gnathostomes with a mouth with a movable jaw. Now, the earliest ones were very similar to ostracoderms in a lot of ways. You notice that they're still armored at the head end. They still have this bony outer covering at the head end, but they had a movable jaw. And the ostracoderms did not last very long once these animals evolved. So the group is called placoderms. So when they appeared, they were much more active predators. They had a jaw. If you're some ostracoderm and you're really uncoordinated when you swim and you're not very fast, you don't have much of a chance for an animal like this. And I should point out, like this one, this is, well, the old name for it was Dianichthes. The new name is Dunkleosteus. That animal right there was up to about 10 meters long. So this is not a small animal. These are actually pretty impressive. The evolution of the jaw is kind of interesting, because if we're talking about craniates, obviously they have a cranium, and there it is. It's labeled skull here, but the technical term for that would be cranium. And then what you're seeing below it, these are all skeletal elements that are supporting those modified pharyngeal slits, because now we're talking about animals that are too big to just breathe through their skin, too big, and they've got too high of a metabolic rate. So they need some sort of respiratory apparatus. So those pharyngeal slits are modified into gills. And because these animals are big, they need some kind of skeletal elements to support those gill openings so it's not just soft, flappy tissue. So those rods, which are not part of the cranium, have became modified to form jaws, and you can see it's from a couple of those gill arches, the structural support. And so actually when you look at, well, like a mammal skull, which would include human skulls, some of it is cranium, some of it is derived, at least evolutionarily, from the skeletal support for the gill arches. So that's and again, we know quite a bit about this because we're talking about hard parts, so this fossilizes fairly well. When it comes to the evolution of modern vertebrates from those early ancestors that we just talked about, there have been a lot of ideas about how that may have happened, and we really haven't had the fossils to back it up until fairly recently. So what you're looking at here is an outdated phylogeny of fishes. So the green ones down here at the bottom, these are placoderms. These are those armored jawed fishes. But we have a problem because they're pretty bony, at least around the head end. You've got a lot of bony elements there. And of course, if you look at living fishes, some of them are bony, like these right here and these right here. These are fish descendants. They're all bony, but there are also some that don't have bones. These are cartilaginous fishes. So there was uh, a lot of debate about, well, how did that happen? Because we have this fossil evidence of pretty bony fishes here that are going way back. So how do we get from there? It kind of seems like, well, it makes sense to go from here to some of the bony fishes, but how do we get from these bony ancestors to cartilaginous fishes? And so there's been a lot of debate about that. Um, exactly how that has happened, like I said, depended on both getting some new fossils and also some uh, getting, doing the right analysis on it. Now, if you look at the way that this is saying this evolution happened, right here, so we've got these bony fishes, these placoderms down at the bottom. The green ones are bony, and then we're saying that that bone is progressively lost and becomes cartilage. Then, and so there we go towards the cartilaginous fishes. So we lost the bony skeleton, and we had cartilaginous fishes are the descendants. But at the same time, another branch, after it lost its bony skeleton, developed bone again, and that's the origin of the bony fishes and the descendants of the bony fishes. Now, so think about that. That is saying that, okay, bone evolved here in these placoderms, bone disappeared here, and then re-evolved again up here, which is kind of a complicated set of evolutionary steps to get where you're going. This is our current phylogeny, and so you see at the bottom, again, we've got these placoderms. This is Intellignathus, which is something, something like a very, very advanced placoderm. And Intellignathus is an actual fossil. We've found them, and that is probably pretty much what the common ancestor to all of the living jawed vertebrates was like. It was either Intellignathus or something very closely related to it. So these are all bony fishes, and you notice in this phylogeny we lose bone and wind up with a cartilaginous skeleton once there. And then if we follow the branch that leads to bony fishes and the descendants of bony fishes, all we're doing is modifying that bony skeleton. It's retained the whole time, and we think that that's probably correct. Again, it may change as new fossil finds are made. You never know.